Yes, good morning to all. So today we may move further in a same unit and topic related to colonial broth culture characteristics. What is in particular? The characteristics means what? Whenever I'm asking, very good, very good. So all the basics features we are going to learn. Characteristics, what are the basic features? What it follows, how it behaves. So in it, here we are going to learn about the two into it, that is in when it is in broth and when it is in a solid medium in a plates. I think in last you have seen that some of you people have shown me the tubes, E. coli tube, having the some on upper layer. It was having them some flocculation. You are showing that whether it is contaminations or not, right? Some people have told that, or I just told that it is contaminations, and I told that it is not a contaminated. It is not been contaminated, right? But it has the sum of when you are going to keep any tubes either in a static condition or in a shaking conditions, their character is going to change. What's going to happen? Like the liquid medium is there. I have one suppose a tube. I'm going to keep them in a static conditions. Then the bacteria start to grow within the liquid medium, right? But it may start to get collect or together. Maybe it is dispersed throughout, or may get to stand at the below, at the butt, or maybe they are on a floating at the near to the surface. Or some are maybe are going to stick near to your tubes, that is on a wall. Okay, they are going to stick on the wall itself. So whenever you are going to get each of the microbes having their different kind of a characteristics. So based on this, some basic features also you can understand that suppose the microorganism what I am looking or growing, it is E. coli or the something else. So it's called the basic characteristics. Some of the when you are going to keep the bacterial solutions or the microbes in a static conditions, they may start to secrete or release some specific kind of a chemicals. They are going to chemicals, or you can say the coloring agents, you can consider as a dye. These are the secondary metabolites for them. Okay, so it is not required for them. It is a metabolically during the metabolic process, certain things are get produced and it is get released out of it. And it may going to stick on this either surface or may get flocculated or get at the bottom, it's going to get collected. Sometimes they may produce like oil-like structure, organic compound over there. So it is not get dissolved. Like oil and the water cannot mix, right? So it is going to get flocculated on the upper layer. So these are the some basic characteristics of any of the microbes when it is kept in static condition. Now the same thing, you have inoculated one tube, okay, and you have kept in the static condition, another tube you have kept in the shaking condition. When you are going to keep the same culture, same medium in a static to in a shaking conditions, you will observe the different kind of a characteristics. Maybe during the shaking, it may be aeration may be higher. We are just generally talking about the aerobes. It can be for the anaerobes even, but just thought like it is any of the organism is there. So they are going to get like aerobic condition is there, more oxygens will be there, right? Oxygen level is going to increase. So metabolically they are going to get active more. As the metabolically they are active, they may start to produce extra material. There will be a cell division, excretory systems will be going to get activated. So there may be some waste product or the other products which is maybe not essential for them, microbes will start to grow and going to release out of it. So tubes were same, medium was same, bacteria was the same, but when I have kept into one was in static and another in a a shaking condition. Now, when I'm looking for the tube after a 2 days or a 1 days, you will observe that maybe they are going to get dispersed evenly because it is rotating. So there is a dispersion. मतलब कि आखा मीडियम ની અંદર એ સારી રીતના છવાઈ ગયો હશે. Okay, it is like a when you are preparing while making the you want to wash your clothes. Okay, so you are taking one bucket with a full or half fill with the water, and then you are going to add the detergent. After adding the detergent, what you are going to do, you are going to mix with your hand or tumbler you are going to use. If you are going to keep the detergent as such at the within it, it will take a day or 1 day or 2 days maybe, that a detergent maybe start to get dissolved and dispersed within the water bucket. Or it may happen that it may form in a gradient. Detergent at the bottom, it start to get dissolved, at the upper layer it will be okay, it start to disperse. The detergent is going to get dissolved within it. So what you have done, you have used your tumbler to mix it well. So within about 30 seconds to 1 minute or maybe 2 minutes, it's going to get dissolved. So it is like a solution is being prepared. Like that itself, here the E. coli culture is there. Maybe it is then shaking condition now. It is going to get dispersed evenly. Okay. Other than that, now what's going to happen? That it may start to release certain chemicals, extra chemicals, or the waste product may be going to start. It may start to disperse in it. So same tube. So on that day, what happened? That first in a half day for overnight, it was in a shaking condition. At the morning time when the Suresh came, he has just switched off the incubator. He has taken out the tubes and kept outside. So it has received the characteristic of both. One was the shaking, then the static condition. So from morning 7:30, and we have reached at around 10:30 or 11 o'clock or 11:30. So about 4 hours that tubes were incubations in a room temperature, but it was in a static condition. So some of you has got the tubes having the flocculation, right? But it was not a contamination, right? It was not a contamination, but it was a flocculation of the same culture. So these are the some basic characteristics, okay, that we have to learn one by one. Okay, so just first related to culture. Other than that, that is in liquid culture. When you are looking for now solid medium, that is on a plate. There could be one plate or on a slant we can go. Slant is produced where? On a tubes, like in a sugar tubes or in a test tube. When you are going to keep in the tilted condition, the agar is going to get solidified in a way that it's going to get the slanting. Suppose it's a tube with me. We are going to produce the solid media in our next class, our next practical. Maybe it is tomorrow. Okay, so we'll ask you people to prepare the solid medium. Just first targeted part will be for us will be a solid medium preparation. Okay, so what you are going to do? You are going to prepare the certain plates as well as certain tubes. You are going to prepare. So within that, what we are going to do? Just fill the tubes. Okay, we are going to fill the tubes with agar. Okay, that is a nutrient agar is there. Then after autoclaving, not before. After the autoclaving only, we are going to take the tubes from the autoclave outer side, and we are going to keep the tubes in a tilted condition. So we have to do such arrangement. Okay, there are the certain stents are also coming specifically meant for that. To all the tubes you kept in the test tube rack, and this test tube rack can be tilted in certain angle. So there is no need to worry about it. Angle will be managed. Otherwise, what you can do, you can keep that tubes near to the certain racks in certain positions. You have to be need to take care that it should not be start to rotate. Just it is kept, and some friend came. Oh, what are you doing? Or let's go for the break or something. Yes, yes. Just happens you are not knowing the tubes were kept. It started to rotate and fall down. Then it having the cotton plug. The media has reached to the cotton plug. So nobody is saying, okay, I'm going to put us in a proper condition, right? Now again, now your experimental part is gone. So one has to be take care about it. That's the you are not going to spoil the media, and while after taking from the autoclave, it should be must be know that it's somewhat hot in nature. Okay, so if you are going to hold the tubes from the bottom, it will be very hot. So hold from the somewhat at the middle of the part. That is 33. Okay, that is the rule called the 33 tubes, 33 holding rule. That whenever you are going to, don't start to that mouth part, because sometimes the mouth part, if you are looking to touch or that part, you may touch the cotton plug or maybe it fall down. So it is the better holding you are going to get proper. Okay, so it is a 33 is a better. So when you started, keep on the slanting condition. Now the second point is going to come, that the bacterial culture maybe you are going to do. Okay, on it. We are going to same plates you are going to store for a week. On the next part, we are going to do a sticking on it. So it will be done in a group wise media preparation. In the next, you are going to do a sticking on it, followed by the then after it is a spreading. So one by one, we are going in an experimental part. Okay. So then we will go for the pour techniques. The pour technique, I will may require the more lectures. Same day we have to do. While pouring, you need to add the bacterial suspension into it. So maybe in a next coming practical sessions, we will follow into it. Okay. So one has to take care about the each stage. Now the second, you had done streaking is done. Okay. Then on a next day, when you are looking to check, we are going to keep this plate on an incubator, and the plates always remain in a static condition. We are not going to keep in the shaking conditions. There is a no need of, because it is already in solid media. Whatever is going to happen is in a static condition itself. Okay. Now on the next day when you are coming, if it is a soil sample is taken, you will find the number of the bacteria over it. If it is the only one bacteria, it's like a broth is given to you, liquid medium is there. If it is E. coli, so you are going to get some specific kind of a over that colonies. Now we are going to learn about the here in this picture you can see the colorful several things, right? So it is nothing but the different kind of a colonies. Each bacteria having their uniqueness for their growth. The group of bacteria may be following like cocci are there, having the similar characteristics. Other having the another group having the somewhat different their characteristics. Some are mixing with one to another. Okay, so what are the basic characteristics are there? We are going to learn about 7 to 8 into it. It is whether it is raised or not. One, the color, whether it is a white color, green color is there. Other thing is whether it is a transparent or the translucent or the opaque. You need to keep the plates below near to the lights is there. Whether it is transparent or not. Then you are going to learn like a rough is there not. Sticky is sometimes. When you open the plate, you may use your nichrome wire and you are going to touch it. You will find that it is oily and the sticky. Some are very much rough. By appearance also you can see whether the plate is sticky. It will be similar, seems like oily content. Okay, so these are the some basic characters in the solid itself. Then their texture and the dimension. Some here you can see some are the round in shape. Okay, some are followed into some different patterns. Okay, there is a irregular shape is coming. Other than that, you may see that the 2 to 3, like a first, second, and third, you can see it having the one orange color, brown color, followed by certain yellow. So same plate, within the one plate, one microbes is exhibiting one particular type of characteristics, where and the another plate, some are the pink, some are the white. Okay, so each bacteria are going to have the different characteristics. Okay, but they are their key characteristics. Once you learn and you are working on the some one organism, then after you will come to know that this kind of a structure or appearance is coming, this bacteria is only present. Okay, it is like when you are preparing some material as food or like that. Sometimes you are find that it having the some, it is get spoiled. But you come to know that whether it is that much harmful or not. Like a bread is there or the stale food is coming sometimes. Okay, you come to know that it is the having the some fungi or the greenish or the reddish patch skin. So you come to know it may be harmful. So you are going to throw it out. So no one is going to say. Otherwise, when you open your uh, tiffins, okay. So if it having the some smell, you will start to say that this food is not of now that much good quality. Maybe it is get by chance spoilage. That is the food spoiled because of some microbes has grown. So no one is coming to tell you that the food is of a quality or what. Or sometimes when you test it, right? Food when you test, you will find that it is not of good quality what you are eating daily. So based on that also you can come to know these are the some characters that food is of a good quality or not. So first is the appearance. Okay, when you are going in there, some a parcel is came to you, right? You have ordered something via Swiggy, something has came. While looking the few points, you can say that today the quality is not good. Okay, either its color or their appearance while the testing. Same certain things are related to colony characters or the broth characters. So many guys or those who are working. Yeah, good question. That is the identification is done first is the morphological and physiological part. We are just currently learning. Second is the biochemical. Biochemical tests are there. I'm not so sure whether it is in this, not in this. Maybe in a next semester or somewhere it's going to come. That the certain biochemical tests are there. That is called the Burgess manual. Have you heard about this? This Burgess manual. That scientist has given the so many tests, biochemical tests, whether it's a fermenter, non-fermenter. Okay, then it is like related to gram negative, positive. So many tests he has given. Based on that, you can confirm that which bacterial suspension is there. That is the biochemical aspect. Other than that, you may go for the molecular based analysis. That is a 16S rRNA. General people are going. That maybe you are going to learn into somewhere in molecular biology related papers will come. The three stages are there. First is there based on their morphology, you can consider. Second stage, you need to go for the biochemical test. And confirmatory test, it is a molecular. At that DNA level, we need to check it out. Biochemical tests are generally going to give assurance 99.99%. Okay. And it is accepted throughout the world. Even the molecular is giving, it is also 99.99%. Okay, because it is the strain level. It may have some variation, okay, because it's a continuous evolution. So you cannot say that it is a stop. The evolution is going on. So always we can say 99.99%. Maybe up to the strain level you can say. Okay, but the next maybe some changes has came. So you can classify in the some particular group of species. 100% assurance is never been came because the science has this their limitations and exceptions. Because it's continuous, microbes are evolving like coronavirus, right? Currently, we are telling that alpha and beta and delta, nobody knows the when it is going to stop. So you are going to say that it's a corona. Yeah, we are going to classify them corona, but now we are going to say it's a D variant. Okay, alpha variants, delta variants. We may going to give this some another name to same strain. Now it is already in a environment. So you may going to say that E. coli. Okay, but E. coli, their strain may have certain change. Okay, that certain change has came into it. So 99.9% you can say. So based on the morphology, those who are working continuously, just based on the observation, you can classify that 90% I can say this one is a pseudomonas only. This one is this kind of a fungi only. Okay, this based on the experience it's coming. Or sometimes just they open. While opening the flask, generally when we are working on the near to burner, so this if the chamber is closed, or if you're working near to the laminar flow, what's going to happen? That while just opening your flask, some smell will start to come. Okay. If you are working on the B mega and others, you will find that you are near to the sewage. Certain bacterial suspension is there. That is a bacillus species. When you are going to near to the open sewages are there, you will start to close your nose. Right? It is nothing but the bacteria will start to grow and they start to release their certain waste product in a form of a volatile compound. And that is why you will just not like the such places. You are moving away from the sewage. So such bacteria, if you are growing on your flask, when you just open it, you will find that a lovely smell is coming that you never liked. Right? So it is nothing but they are. So you can, it is become their characteristics that this bacteria is going to give this kind of a smell itself. Some bacteria are never going to give any smell because they are not going to release any compound. Yes, but other than you can perform in different way. What the medium you have provided, it is a nutrient medium. Right? Earlier I have told about the several selective media, enrichment media, natural media, synthetic media. Okay, so natural media is what? And the synthetic media? Natural, it's all the compound like yeast extraction we have prepared. Okay, in the synthetic media, we are going to add all the elemental parts. It is mixed totally. Here it is, the yeast extract is there. Beef extracts I just told earlier, right? We are going to add the many other elements like NaCl. So it is become synthetic. Natural medium is what? Like earlier, people are used to take the either milk or they were used to take the coconut water. Coconut water is rich in the minerals. So people were using such thing. They were adding the certain different kind of flour into it. Wheat flour, corn flour, they were using. So it is totally natural. We don't know about exactly all the aspects. All the nutritive capacity we don't know. So it's the natural. When you're going to synthetic, you are knowing some basic points. Okay. That each extract, you have added the NaCl, this much of this pH maybe you are going to maintain. So all the thing is being added. So natural media, synthetic media. So while working with some particular kind of bacteria and you are looking that I want to test their particular characteristics, what you can do, you may add some particular kind of a compound within it. So we know that bacteria is going to take it. When the bacteria will start to grow, it's going to release some particular kind of a compound out of it. It is like enzymatic action is going on. You have added the substrate, catalysis is done by the microbes and the product is generated. So either product you may go for the chromatogenics, color change will be there. Another you may thought like it should be start with producing some volatile compound. So volatile compound is there. When you open it, while opening, you will observe that some smell has come. Other than that, sort in Burgess maneuver, when you are working, while preparations of your tubes, some strips are being put inside. It is like some papers. May I have your notebook? If I take some paper from it, you will never mind. Thank you. So you are being advised to do certain strips preparations. What is being required to do? That if I open the tubes, right? If I do have the tubes, what I am going to do? I'm going to put within inside. Half of the part, maximum part is lying inside, and something is remain upper part. So when I'm going to close it, so what's going to happen? Some paper has moved inside. Here it is not a tube, it's not going to happen, right? So these are the coloring and the chromatogenic material. Either you go for the pH strip. If certain bacteria, we know that they produce some acids. Okay, if the acid production is there or the chromatogenic volatile compound is being produced, it's like a chemistry now. You're working in the chemistry lab, right? You know so many things over there. You have blown it, maybe you're doing some blowing and everything, right? After the blowing and making the hot, color change, appearance change, texture change. Same thing is you are going to do over here. You are going to keep the tubes for a week or for 1 day. The color of the strips within that is changed. You can say that this is the basic characteristics of my microbes. Okay. Other than that, we are going to use some Durham's tubes. Maybe you are knowing or not heard Durham's tubes. Okay, no problem. Durham's tubes are very small. Small vials are maybe you can say about two maximum 1 or 2 sorry 2 to 3 centimeters and very thin. Not a append of tube. It's the make of a glass. Same similar like a test tube. Small test tube, very small test tube, very thin, very small. Not a append of tube. Glass make. I'm going to show it. So what is required to do? The tubes are put inverted in the place. It is like a suppose it's a tube. Yeah. If I'm going to cut it, not right now again. Earlier I have cut the one of the students, right? So it seems like this. It seems like a tube now. This is the Durham tubes made up of a glass. Just it. Okay. So it has been identified, prepared by the scientist Durham's. So it is known as a Durham's tubes. What is been required to do? You have to put in inverted conditions. Means top. Okay, bottom is coming at the upper part, and you are going to put. It is like a tubes with me. Okay, tube is there. It is liquid is here. Okay, what I'm going to do? I'm going to slide it down. So now it is within the tube. I'm going to insert another tube. Okay, it is been fed inside. So what's going to happen? That after you are going to just rotate for a while, it is going to fill with liquid. It is going to get filled with the liquid. You have kept for a day. When you are coming on the next day, you will start to feel like that half is filled with the air. You will surprise who has filled the air. So sometimes it may start to, because of the air pressure is very high, glass tube is start to float even somewhat because the air is making a pressure over there. So you'll surprise that, oh, it's a magic, right? It's a kind of a magic because when you add it and it was in inert conditions, even though liquid has moved inside, because it is having the thickness is so much in a way that it is going to move inside the water. Okay, if it is not there, just shake it. It should be get filled with the your liquid medium. Air should be not there. Just shake it, it will move. Now it was there within inside. Next day you will realize that it has filled. So what was there? The bacteria has started to produce certain gases. Okay, so bacteria were inoculated within this plate. They will start to produce some gas. Sometimes you are when you are here we are having the one some lake, right? Whenever you're going, or sometimes you are knowing here and somewhere wherever I don't know exactly. Okay, you'll find that sometimes the bubbles start to come. As such, no one has touched anything, but bubble is coming. From where it's the bubble is coming? You are telling the algae is doing something experimental part. If you are going to remove the algae, something is also going to happen. Yeah, the bacteria. So they produce this thing. So this will be the another characteristic. In a liquid medium, they will produce some gases, and this gas is going to fill. So it is a experimental part. So you are going to club a chemistry, knowledge of a chemistry in microbiology. Okay, this is the basic part is there. You are going to use your chemistry. That is why for the microbiology student, you are going to learn the chemistry. I don't know, no one has told about it that or not. Okay, what is the importance of such paper during your course related specialization in microbiology? You should able to learn organic and inorganic certain properties. You right now maybe not knowing, but when you're doing certain experimentations or the research at that level, you will realize that I know a certain principle of the chemistry that can be utilized here. So this is the combination of a two to three things. We are going to learn some analytical techniques. So the physics also coming. The certain laws are coming. That's based on the physics. So you are not going to learn the physics properly in more detail, but it will be a biophysics, considered as a biophysics even. So such basics part are coming. So basic important part is what? Characteristics related to the liquid medium, characteristics related to the solid medium. Now again, On a solid medium, when you are working, you come to know certain options are there. Okay, there also you can use the solid medium in a way that you can differentiate. I have just told about the differential media, right? Mecon keys agar, eosin agar. So we are going to use the when eosin agar or the fermenter, non-fermenter classifications are there. So already you maybe have added some chemical chromatogenic agents over there. Dye is been added. So on the next day when you are going to come, plates going to appear as the some different colors. Dye color means plates color is changed. So you can say that this microbes is only present, or that this group of microbes are only present within it. Then you want to confirm again, we need to go for the biochemical test. That is the next thing. Other than also you can do. You can have the plates, nutrient agar plates were there. You may just open the plates and pour or flush with the certain chemicals. Dye or the certain chemicals are coming. When you flush it, it start to have the some reactions into it, because already organism has produced some particular kind of a chemicals. They were lying outside. When you add something into it, the color change may happen. Like you are working phenolphthalein and others, whether it is some compound, just one drop you are going to add, your pink color solutions became colorless. Happens. So many experimentations are coming in chemistry. You are doing some estimations, I hope. Estimation is coming, some burette is there. You open the core. Titration, yeah. Titration. So you are going to reach the some stage and steps. There you are going to say, okay, I reached this place. The titration is done to come to know when it's going to get saturated. The same saturation point you already noted, and that you are going to use in your flash. That if I'm going to flush with the 2 ml of my solution in the bacterial colonies of or of a such number of them, it must has produced this chemical in this much amount. If I'm going to flush my plate or the floor over this and keep for a while, just a plate is been opened. Okay, plate is been opened in the sterile condition. You flush it and just with using your palm, keep on the platform. Within few seconds or minutes, you will see that color change will start. Either color change will happen, bubbling will also going to happen. Depend upon the bacteria, what the chemical you are going to add. So the reaction of the chemical that been produced by the certain bacteria, it's going to appear over here. So these are the called the some techniques to identify or the classify the bacteria what are present. So when this kind of activity is also done, sometimes you come to know totally new some microbes or pathogen is there. So we need to understand them at their basic level. So first observation is coming with their medium on their plates. If on the plates you are going to find that this kind of a colony is there, it must fall in this kind of way bacterial population only. Okay, so accordingly we need to give the medications into it, right? So pathogens are there, how to identify, or the normal bacteria are there, how to identify. Okay, so this is, I'm going to go in a detail. Till now, anybody having any questions related to this? We are going to learn each step more one by one in detail. Okay, just I have given the overview about the topic. Anybody has any questions? No? Okay. Yes, before this, in unit number 2, okay, this one is, I think, number, let me check. Yeah, 2.2, up to 2.2, 2.2A, B is going to cover. Right, so let me show. Yeah, yeah. So methods of isolating pure culture that we are going to include over there. Yeah, just I mentioned. Okay, now days what? Yeah, 11th August. So yes, selective method for bacterium selections. That just already I have discussed, but I'm going to take that point maybe next part. Okay, methods for culturing aerobic and anaerobic bacteria. That already I just talked. 4 plates, 6 plates methods are there. So till 2.1 and 2.2, it's going to cover from the unit number 2. While related to unit number 1, yeah, nutritional types, requirement of nutrients for microbes. 1.1 is going to cover. Then 1.2. Over there, I have taken up the photoautotrophs and photoorganotrophs. Okay, up to 1.2, I have covered till this. We are not going to learn about this chemolithotrophs and others. So that you have learned about oxygenic and anoxygenic. That pathway I have told. Till it will be going to cover. This chemolithotroph and chemoorganotroph, we are not going to cover over this. Yeah, definitions and that part I have already told. That chart and tables I have discussed. So it is can be as the define the chemoorganotrophs or chemolithotrophs. But the detailed mechanism of their transport or the ATP generations, energy generations, that is not going to cover in this mid-sem exam. Okay, and yeah, I think Aditya sir has covered this primary and secondary transport. So transport mechanism, that already the same topic is there. So it has been decided that he is going to take it. It is covered. Yes. नहीं आखो न था तो. This one is the, you can say, to understand this point, it will require what just I told earlier even in whole paper. To understand some phenomena or the pathway is most important. Okay, so very difficult to understand sometimes, or you need to give your more attention over there in just 2 points: chemolithotroph and chemoorganotroph. Right, just 2 points are there, but it will take a lot time to make you understand. It is like a similarly one unit you are learning. As per my just thinking, I'm telling that it will be a more weightage is generally given because of out of it, one questions always will be there. Okay, there is a 5 to 6 categories are coming into falling into this. So at least over 6 to 8 marks is going to cover maybe in the next whenever it will be there, because it having the some pathways that you have to learn into it. So almost you can say, but not full units. Okay, so the transport and diffusions, that part is already taken by him, or it's completed. Aditya sir, transport mechanism, primary and secondary active transport and diffusion. I think he is taking. So it is there. So one unit number 1 into it. 1.2, 2 points are, we are not generally just going to take over this right now. Okay, that will be covered later. Okay, and the unit number 2. Okay, directly in unit number 2 till 2.2. Anybody having any other questions? I have also uploaded, right? 1.2, 1.2a, 1.2b, 2.3. Okay, that classifications I have made. Accordingly, I have uploaded in Google Classroom. Same thing is there. You can find the links. If I have taken the 1.2 in a 3 lectures, I have written as a part 1, part 2, part 3. on a YouTube. Links are there. In a Google Classroom, you can check it out. So already I classified each point is there. Just maybe I have written the 1.2. Okay, there I may have classified 1.2a, 2b like that. So 2a, 2b. So 2b, if I have just covered, or 1.2a is there, I have devoted maybe a 3 or 4 lectures. So in YouTube video, it is written as a 1.2b or 2a, part 1, part 2, part 3, part 4. Okay, so you just check it out and start to prepare. Maybe it's on 24th, right? So long time is there. That is overnight, right? The learning is always overnight, or it's daily. Depend upon the guys. Okay, some people are doing the daily, some are on the last day runners, some are coming just to enjoy the life, just to check that the sir is asking what sort of the questions, how I am replying and responding, right? But it may be must be offline. I hope it is offline. Those who are in online mode, they have to come in offline format also. Okay, we are going to keep some social distancing. That will be taken care by the university and college. The guys are there, so it will be not your headache. Okay. Yeah. Other questions related other than this mid-sem? Anyone? No questions? Any questions related to this or the further? So right now I'm not going to continue. Okay. You can have your. Is this? Yeah. Right. Going to put this attendance link. Put your attendance.